And first up is Rachel Moran.
Okay, thanks so much for the introduction earlier, and thank you for having me. I'm very honored to be here presenting in. Okay, very honored to be here presenting in the ASN. Okay, in the ASN Early Career Investigator Award Symposium. Today I'm going to talk about mechanisms and consequences of repeated evolution. So a central goal of evolutionary biology is to understand the connection between phenotype, genotype, and fitness. And we now have some really nice examples where variation in adaptive phenotypes have been mapped down to underlying genetic variation, in some cases down to specific genes. And systems where the same traits have evolved repeatedly across independent lineages have been really useful in identifying connections between phenotypic and genetic variation. Repeated evolution is often associated with really strong selective pressure, and traits that have evolved in this manner are often adaptive. There are some really cool examples of repeated evolution across deep evolutionary timescales, like the evolution of wings in bats, birds, and insects, and the evolution of seasonal coat color changes across birds and mammals. But examples of repeated evolution within closely related species or populations within species, like these marine stickleback that have repeatedly invaded freshwater systems and adapted to those novel environments, can be more useful in helping us to understand how evolution proceeds given a similar starting point and similar underlying selective pressures. And these systems can also teach us about the predictability of the evolutionary process. So in my work, I'm really interested in local adaptation and speciation. And to study these processes, I use two different groups of North American streamfishes. So today, I'm going to start out by talking about some of my more recent postdoctoral work on the genetic basis of repeated adaptation to caves in Mexican tetras, shown on the left here. And then I'm going to switch gears and talk about my dissertation work on the repeated evolution of behavioral isolation in darters, two species of which are shown on the right there. So in the first half of my talk, I'm going to be focusing on the mechanisms of convergent evolution. And then in the second half, I'm going to talk more about the implications. So there are several ways in which repeated evolution of the same traits can happen at the genetic level. We might see that the same genetic changes occur repeatedly across replicate lineages that have evolved this same phenotype repeatedly. So here, these black arrows going from the ancestral to the derived phenotypes are indicating that the same underlying genetic change has occurred repeatedly. And alternatively, we might have variable genetic changes occurring and leading to the same phenotype across replicate populations here. When we see a scenario like the one on the left, the simplest explanation might be that gene flow has occurred and passed those adaptive alleles across those replicate populations so that it looks like the same underlying genetic change might have occurred repeatedly. But we could also easily see this type of pattern if selection has occurred from standing genetic variation. And with the scenario on the right, the simplest explanation might be, or the most assumed explanation might be, that there are de novo mutations that have occurred across each of these replicate lineages leading to the same phenotype. And there are many factors that might influence the mode of convergence, so this can be quite complicated. And we might be more likely to see a scenario like the one on the left if there are limited genetic mechanisms for producing a given phenotypic change. So if we see more genetic constraints, and we might see fewer genetic constraints or pleiotropic effects if we see a scenario like the one on the right. So identifying the genetic underpinnings of phenotypes that evolved repeatedly can provide some insight into the mechanisms that promote adaptive evolution. Astyanax mexicanus, the Mexican tetra, provides a really excellent evolutionary model system for studying repeated evolution. So in the Mexican tetra, we have ancestral surface lineages that have repeatedly invaded caves and experienced convergent evolution in the same suites of traits, including morphological traits like loss of eyes and pigment, metabolic changes like increased fat stores, resistance to starvation. Caves are really dark and there's not a lot of nutrients in caves. There have been a lot of cool behavioral changes like loss of schooling and aggressive behavior and reduced sleep and disrupted circadian cycles. So these fish don't sleep a lot because they're fish that are sleeping less are spending more time foraging, so this is thought to be adaptive. And so all of these traits have evolved repeatedly in this system, and a question remains as to what order these traits evolved in upon initial entry into the cave system, and does this order of trait evolution vary across lineages? Which is one question I'm interested in. I recently did some phylogenomic work that I don't really have a lot of time to go into, But I'll just mention that there are about 30 caves present in northeastern Mexico where the cave adaptive form of Astyanax mexicanus has been identified so far. We have the Guatemala cave populations for this north, then the Alhambra cave populations, and the Micos cave populations. Some of my recent work has shown pretty definitively that we know that we have a southern and northern lineage surface stock that moved into northern Mexico and then subsequently invaded 2 different cave systems, giving rise to distinct lineages of cavefish. And I refer to these as the southern lineage with the Alhambra caves and the northern lineage, which contains the Guatemala caves. Micos cave populations are pretty complicated. They have a complex history with lots of hybridization, so I'm not going to talk about those ones today. Okay, so 2 questions that I wanted to ask with the system or to answer with the system were, which traits were most important in facilitating cave adaptation? So do we see a morphology, physiology, or behavior first type of scenario with adaptive evolution? And then what is the mechanism of convergent evolution? So is convergence between lineages proceeding due to selection on standing genetic variation, gene flow, or novel mutations? Here is a really quick overview with the flowchart of the steps that I took to answer those questions. I'm going to walk you through each of those in detail, but just briefly, we sequenced a bunch of genomes for cavefish and surface fish from multiple different populations across lineages. I then identified selective sweeps in cavefish genomes and also looked at surface fish and looked for regions where there was a sweep in cavefish and looked like neutral evolution in surface fish. I filtered down genes within those regions to only retain those with Go terms of interest, so those related to known cave-derived traits. And then I dated that subset of selective sweeps to try and get an idea of the ordering of when those selective sweeps occurred relative to one another and across these different Go categories. And I then finally inferred the mechanism of convergent evolution in these adaptive alleles across lineages. Okay, so let's start off with some of the sequence data. So all this cavefish work I did as a postdoc in Suzanne McGaw's lab at University of Minnesota. And this was all highly collaborative work, and this was work of lots of different labs working together. And so we sequenced 257 genomes from 18 cave locations, shown with stars up here on this map, and 8 surface locations. And using this data, I then identified putatively adaptive alleles, so genes that had a sweep in the cave population and what looked like neutral evolution in the surface population. And I used a machine learning approach in Diploshic to do this. Happy to talk more details about that later if anyone is interested. And then once I had this catalog of selective sweeps that were present in cave populations and then neutral evolution happening in surface populations, I identified genes with Go terms associated with known cave-derived phenotypes within those sweep areas, things like eye development, pigment, or sleep. And I did this in a total of 3 northern lineage caves and 4 southern lineage caves that we had good population sizes and good genomic coverage for. And we can see that there are about 172 of those genes that overlap between the lineages, suggesting convergent evolution between these lineages. And the next step was to then date these selective sweeps. So I used the genealogical estimation of variant age, GEVA, which takes a coalescent approach to determine the relative ordering of these selective sweeps within each cave population. Here I'm showing the results for just one population, Molino. So on the y-axis, I have the sweep age, and this is generations before present. So as you're going up on the y-axis, you're going further back in time. And then along the x-axis, I have these different categories like eyes, metabolism, neuromasts. And sort of reassuringly, what we saw with these results are that when we look at some of our demographic models and inferences into previous population size changes, we know that the caves experienced a bottleneck corresponding to when they split from the ancestral surface lineage about 150 to 250,000 generations ago, shown in this gray vertical bar here. And that corresponds really nicely to when we are seeing these selective sweeps pop up initially across the cave populations. But interestingly, we are not seeing any difference in sweep ages across phenotypic categories. Okay, so first I was like, oh, well, that kind of stinks. But so what our sort of working hypothesis is that this cave, the cave environment is just really harsh and it has these really strong selective pressures. And so we think that basically everything is under selection simultaneously and that evolution is highly pleiotropic, highly polygenic, and happening really rapidly. And there are many pleiotropic genes that are popping up in this analysis as well. Yeah, and so we see this, some other recent work in the cavefish system is suggesting the same. Okay, and so I also wanted to ask what the mechanism of convergence was among these cave lineages. And here I'm going to focus on presenting data from what we sometimes call the three core populations, Molino, Pachón, and Tinaja. These populations have been central to a lot of past work in this system, and they have well-maintained lab populations. Molino is up here in the Guatemala region, and then Pachón and Tinaja are in the El Abra caves. You can see the Pachón and Tinaja are, although they're both within the El Abra cave system, they're quite geographically distant from one another. And so I was able to look at signatures of convergence between and within lineages. And I identified 760 genes that had overlapping sweeps in all three of these caves in neutral evolution and surface populations. And I considered those candidates for convergent evolution. And then I looked at a sort of a control comparison where I identified 172 genes that had sweeps across three surface populations, two northern lineage and one southern lineage, and neutral evolution across these caves. Okay, I then used this approach, distinguishing modes of convergence described by Lee and Coop in 2017, to try and investigate the underlying modes of convergent evolution in those candidate genes. And so this method, again, uses a coalescent theory to model neutral evolution and several alternative models of convergent evolution, including independent mutations, standing genetic variation, and migration or gene flow. And it uses population genetic data. So here are some results. So the blue bars are showing surface convergent sweeps, and the gray bars are showing convergent sweeps across the cave populations. And the first thing that sort of popped out was that 95% of these genes, so genes, the proportion of genes are shown on the y-axis, and then our three modes of convergence are along the x-axis. 95% of the genes in surface fish that are candidates for convergent evolution appear to be evolving due to selection on standing genetic variation. And 53% of genes in cavefish are the same, also evolving due to selection on standing genetic variation. But when we look over at the independent mutations category, we see that they play a much larger role in convergent evolution in cavefish compared to surface fish. So de novo mutations are key in convergent evolution between cavefish lineages. Another interesting result that came out of this was that when we look at these genes that are evolving convergently, and we look at the length of these genes, here I'm showing the transcript length along the x-axis on these density plots. The whole genome is shown in red in both of these plots, and then on the left we have the cave shown in gray, and then the surface convergent sweeps overlaid here in blue. And we see that genes evolving convergently are longer in cavefish, and if we break this down by mode of convergence, independent genes that are evolving via independent mutations among lineages are really driving this pattern. Okay, so this suggests that having a larger landing pad, so to speak, allows for a greater chance that you're going to get a beneficial mutation pop up multiple times, and that longer genes might be more likely to be the target of repeated evolution of adaptive traits. Okay, so what I've shown you so far is that the age of selective sweeps across categories of cave-derived phenotypes does not vary within cave populations. So rather than adaptive evolution happening in temporal stages, the harsh cave environment may have imposed these really strong selective pressures, causing these different suites of traits to evolve simultaneously. And we're seeing that evolution is polygenic and happened really rapidly. We're also seeing that selection on standing genetic variation and de novo mutations drives convergent evolution of adaptive alleles among caves. And so this work supports a trend we're seeing from the literature showing that de novo mutations may be more common in cave populations compared to surface populations for multiple different reasons that I don't have time to go into right now, but that's super interesting. And it also lends support to another trend in literature showing that longer genes might be more likely to be the target of repeated evolution. So overall, this has provided some new insight into the evolutionary mechanisms underlying repeated evolution. And some of the next steps in the system are to use CRISPR to do some functional validation of some of the candidate genes I've identified, and that's all being done by Joanna Kowalko and her lab, a collaborator of ours. So stay tuned for that. So the prevalence of de novo mutations in convergent evolution among cavefish lineages highlights that there can be many unique genetic changes leading to the same phenotypic change. And I think an interesting question that comes from this observation is, what are the incidental consequences for species diversification? Okay, so switching gears a little bit, I'm now going to share some of my work on speciation in the darter system and tell you about the evolutionary consequences of repeated evolution of behavioral isolation in these fishes, And then finally, I'll take a step further back and talk about the implications for understanding macroevolutionary patterns of species diversification. So biologists have long appreciated that mate choice and competition can promote diversification within species and lead to speciation. And it's now clear that these interactions also commonly occur between members of different species, which we call interspecific interactions, and this can have major ecological and evolutionary consequences, one of which is character displacement. So here I'm showing, so character displacement describes trait evolution and sympatry that's resulting from interspecific interactions. Here I'm showing this range of species 1 on the left and the range of species 2 in orange on the right here, and they have an area of range overlap and sympatry. On the right, I'm showing these trait frequency distributions, and in the allopatric plot here, you can see that these species overlap almost completely in that trait distribution, and in sympatry, the trait value has been displaced. And so this is sort of the hallmark geographic pattern that we look for when we're trying to diagnose cases of character displacement. We get this exaggerated trait difference between species in sympatry, and this can act to prevent maladaptive interspecific interactions. So anything with a negative fitness consequence for either species. For example, mating traits can diverge to prevent maladaptive hybrids from forming. And there's 2 different types of character displacement that I'm going to talk about today. The first is reproductive character displacement, and as the name suggests, this involves selection to avoid interspecific reproductive interactions. And so you can have species diverging in mating traits and sympatry. And with agonistic character displacement, we have selection to avoid interspecific aggressive interactions. And you can have species diverging in competitive traits, things like access over mates or territories. My dissertation research used darters as a study system to examine when we can expect these phenomena to occur. Darters are a highly diverse group of North American stream fishes. I believe they're the most diverse group of vertebrates in North America. There's over 250 species, and they each exhibit unique species-specific male nuptial color patterns or ornamentation, like these egg mimics shown here. Darters are a great system for studying interspecific interactions because they can co-occur in these really diverse communities of up to 12 darter species within a single river drainage. Hybridization is also rampant, even among quite distantly related species. You can find hybrids formed, and males are highly aggressive towards other males. And it was initially thought for a long time that female mate choice on male color pattern was driving speciation in this group, but we're increasingly seeing that male mating preferences and male competition are really important in driving speciation and diversification in darters. So my research focuses on 2 wide-ranging groups, the orangethroat darter, shown in orange here on this range map, and then the rainbow darter, shown in blue. These species have similar male color patterns, so they have red and blue banding on their bodies and fins. They have similar ecology and mating behavior, and they commonly hybridize in these large areas of sympatry where they occur in the same drainages in the Midwest and the Ozarks and the Eastern Highlands. And I believe they are the most commonly hybridizing pair of darters. An interesting thing about this system is that the orangethroat darters have been split over just within the past couple of decades into what is now considered a clade of 15 allopatrically distributed species. So that's a lot of orangethroat darters to keep track of. Some of them occur within only one river drainage, like the strawberry darter in the Strawberry River drainage. And notably, 13 of these species co-occur in sympatry with that other species, the rainbow darter. Another really interesting thing I think about this system is that previous work by Mark Pyron and work led by my PhD advisor, Becky Fuller, and a previous lab mate, Muchu Zhao, showed that within species, females do not prefer more colorful or larger males, and that in at least one sympatric river drainage, they appeared to have no preference for conspecifics versus heterospecifics, which seems really crazy. They have traditional sex roles and this striking sexual dimorphism. These are females shown on the bottom here and males up on the top. But some of this previous work also suggested that males might exert preferences for females of their own species in at least one sympatric population pair. So upon starting my dissertation, I was totally hooked by these fish. Sorry for the bad pun. And I thought this was a super intriguing system. I wanted to try and figure out what was going on. So I conducted a bunch of behavioral assays in the lab to measure male mating and fighting biases towards conspecifics versus heterospecifics in sympatric and allopatric population pairings between orangethroat and rainbow darters. So I first did assays for reproductive character displacement, And this predicts enhanced male preference for conspecific females in sympatry compared to allopatry. And then I did assays for agonistic character displacement, and this predicts enhanced male biases for fighting with conspecific males in sympatry relative to allopatry. And I conducted these assays in 6 replicate population pairs, 4 sympatric and 2 allopatric. And here I'm just going to jump into some results. So to orient you here, on the y-axis I have this mate choice with increased preference for conspecifics as you get up closer to 1, and then increased preference for heterospecifics as you get closer to -1. At 0, there's no preference for conspecifics over heterospecifics. And I'm going to show these 6 replicate population pairings here. The black bars are allopatric, where they've never seen each other before, and then the white bars are sympatric, where these species naturally co-occur. And what we see is that there's really strong male preferences for mating with females of their own species, and they tend to ignore females of the other species if they co-occur with one another in sympatric drainages. We don't see this in allopatric populations where they haven't seen each other before. And so this is this nice repeated pattern of reproductive character displacement is happening in the system. I also looked at male aggressive behaviors, things like males will flare their fins at other males when they're trying to get access to a female or do chases and nips at the other males. So I measured metrics of male aggression, and we see again that males bias aggression more towards conspecifics and sympatry relative to allopatry. But what we see again when we look at female mating preferences, that there's no evidence of female preference for conspecifics over heterospecifics regardless of sympatry between orangethroat and rainbow darters. Okay, so what I just showed you is that males show a pattern of character displacement in these reproductive and agonistic behavioral biases, and this seems to be present across replicate populations across the ranges of these species. This pattern is really widespread. And so that's cool. That was a really interesting example of character displacement and male behavioral traits. But for the last part of my talk, I'm going to tell you a little bit about the large-scale implications of these types of repeated instances of evolution of character displacement and what this means for species diversification. So earlier I showed you this really simple cartoon model of character displacement where we have these 2 species with continuous ranges, but this isn't always what species ranges look like out in the real world. So we might see one species where there's more of a patchy distribution or lower gene flow among populations within species, and maybe they coincide with another species that has a more continuous range. So this model is sort of more similar to what we have with the darters, with orangethroats being more patchily distributed and less gene flow among drainages. And rainbow darters actually can traverse larger rivers, and so they have more gene flow among populations and haven't been as heavily split. And so recent theoretical and empirical work indicate that when gene flow among populations is low, this can cause some populations to diverge in one direction due to character displacement, and other populations might randomly diverge in other directions in trait space. And so this can effectively lead to not only differences in these traits due to species 1 and species 2, but also variation in this trait among populations within a single species as this correlated effect of character displacement between species 1 and species 2. So we're seeing convergent evolution of character displacement among isolated populations, leading to behavioral isolation and trait variation among populations within species. This newly described or somewhat recently described phenomenon is termed cascade character displacement, and this has been shown to contribute to behavioral isolation among populations within species in a variety of different taxa over the last couple of decades. And this can sort of act as a speciation pump. So what we mean by that is that it can simultaneously finalize speciation in sympatry due to character displacement between species 1 and species 2, and it can incidentally lead to diversification, initiation of speciation in allopatry among populations within species. And so you can imagine that through this process, populations could diverge to such an extent that they might be considered distinct species or clade of recently diverged species. Okay, so I wanted to ask whether character displacement between orangethroat and rainbow darters has promoted speciation within the orangethroat clade via cascade character displacement. I again conducted a bunch of behavioral assays, but this time I paired orangethroat species that co-occur with rainbow darters or orangethroat species that do not, so those that have experienced character displacement or those that haven't. And predictions for cascade character displacement are that those orangethroat species that happen to be sympatric with rainbows will have really strong preferences for mates from their own population of orangethroat darters and reject closely related orangethroat species. And I looked at this in three pairings of orange-throat darters that are sympatric with rainbows and one pairing that is allopatric from rainbows. And we do see that this seems to fit our predictions for cascade character displacement. So we see that males have higher preferences for members of their own species and have become super choosy and reject mates even from another closely related orange-throat species. We see similar patterns when we look at agonistic character displacement, agonistic traits as well. And this was interesting because it was the first instance showing that agonistic character displacement can have these cascading effects on speciation and allopatry. So just to summarize what I showed you there, we see reproductive character displacement happening repeatedly between orange-throat and rainbow darters, and this has had cascading effects on speciation within the orange-throat clade. We see similar patterns looking at agonistic traits, but looking at female preferences, we see no evidence of female choice between or within species. Okay, so just some conclusions here. We're seeing that interspecific interactions drive macroevolutionary patterns of diversification. And local biological community can influence species diversity through the scenario where increased local biodiversity is promoting further biodiversity. And we know that species are coming into secondary contact at increasing rates due to rapid environmental changes and human activities. And so understanding the ecological and evolutionary consequences of behavioral interactions between species can be really important for conservation, but is often overlooked. So these types of interactions can have really broad implications for invasive species management and species of conservation concern. So just super briefly, I'll talk about some future directions. So I think that cascade character displacement provides a really compelling mechanism to connect processes that shape species distributions and generate biodiversity across micro and macroevolutionary scales. But a lot of fundamental questions remain regarding when and why cascade character displacement occurs. So one of my goals here that I'm working on currently is to try and look at this through the lens of repeated evolution and ask whether the same or unique genetic mutations are implicated in character displacement across replicate sympatric darter populations, and if we see unique mutations or unique traits or genes or gene networks being targeted in different replicate sympatric population pairs, and how this might be contributing to cascade character displacement. And we have some nice reference genomes for multiple populations of both species and lots of PopGen data to do this. And then just finally a plug, I'm opening my lab at Texas A&M in January of 2023, so if any of this sounds interesting to you, come find me and I'd be happy to chat more. And I'd like to thank Becky Fuller and lab and Suzanne McGaw and lab and cavefish collaborators and everyone who helped with this work and funding sources, and I think I have some time for a quick question. Thanks.
Yeah, very nice talk. So going back to the cavefish, did you see differences between regulatory and versus structural genes? And also, did you compare lengths of the genes that were common between the two versus those that were unique?
So we are starting to look at whether or not there are regulatory. So it seems like more of the selective sites, so with this model, we looked at what site seems to be the most likely one under selection, and a lot of them do seem to be outside of coding regions, interestingly. And we did find a few interesting losses of transcription factor binding sites in some of these genes, and we're going to do CRISPR on them, and so that will be really interesting to see what comes of that. There's some really excellent undergrads working on a project related to that. And I think I need to move and switch here, but I can answer your other question after this, because I don't want to take up someone else's time. But thank you.
I think she's leaving.